Good day, dear students. In current lecture, we will continue our discussion of enteric pathogens. We will talk about second genus of enterobacteria. It is a Shigella. Shigella, as any other enterobacteria, is a gram-negative rod-shaped bacteria which is non-sporing, non-fastidious, facultative, anaerobic. In slides, it can appear as a short rod, present as a monobacillus in clusters, and it is non-motile and non-capsulated. According to recent genetic studies, it was identified that actually Shigella and E. coli are very similar genetically, and according to recent data, Shigella can be suspected as some biotype of E. coli, and maybe very soon we will have reclassification of the genus, but still nowadays it is the single genus Shigella within Enterobacteria family, and it has 4 major species: Shigella dysentery, Shigella flexneri, Shigella boydii, and Shigella zonae. To describe pathogenic properties of Shigella, we need to remember that it is still gram-negative bacteria, and it still have the same lipopolysaccharide in the structure of the cell wall, which is acting as O antigen, and have ability to reduce binding of antibodies to bacteria, protects against phagocytosis, and when released into the systemic blood circulation, it's acting as a toxin. Bacteria is non-capsulated, but it have an analog which is called a slim layer, and in serological studies as well, it's acting as a K antigen. Functions are the same as for capsule: protection against phagocytosis, protection against complement activity, and helps bacteria to survive during our immune response. Bacteria have a lot of fimbria which helps to adhere to surface of epithelium and acting as a colonizing factor. Bacteria can release important enzyme which is known as mucinase. That enzyme helps to destroy a protective layer of mucin and mucosa in our intestine, and helps to fix to enterocytes surface and to damage enterocytes. Pathogenic Shigella have plasmids, additional plasmids, which are coded specific proteins which helps bacteria to invade in the epithelium and break down our epithelial surface, epithelial barrier. And as well, bacteria can release enterotoxin, which by structure and functions resembles LT toxin of E. coli, have the same activity, and leads to unique manifestation of shigellosis as diarrhea. Some strains can produce as well Shigatoxin, which acts as a cytotoxin. It can recognize enterocytes, myocardiocytes, and neurons, blocks synthesis of protein, and lead to complete destruction or death of affected cells. As for any other enterobacteria, preferred culture media for cultivation of Shigella initially is Maconchi agar. That agar have lactose as the main sugar and indicator for production of acid, and actually the primary task of Maconchi agar is to suppress other bacteria other than enterobacteria, microorganisms, and only enterobacteria can grow on that culture medium. And 2 main species of enterobacteria is Shigella and Salmonella, which are lactose negative. Almost any other enterobacteria is lactose positive, and initially the task of that Maconchi agar is to differentiate E. coli, which is present in our intestine in large amount, and Shigella Salmonella, because they are much more dangerous and more significant in epidemiology. So if you remember that E. coli colony was pink in color and agar even more pink, if it is Shigella, colonies will be colorless, culture media is not changed, and colonies are absolutely transparent and very small because bacteria is weak in the growth and kind of lazy bacteria. We have multiple selective culture medium, only 3 are included in your program. It is XLD agar, Shigella Salmonella agar, and HE agar. All 3 culture medium helps to differentiate Shigella and Salmonella. Shigella will appear as a colorless, transparent, or very pale colony without any changes in agar, while Salmonella will give you bright black colonies on all 3 culture medium. All culture medium contain inhibiting factors which will suppress the growth of other bacteria, and if something is present, mainly it is only Shigella and Salmonella, but some exceptions are possible. Clearly, iron agar, which should be used for differentiation between any enterobacteria, should be used for identification of Shigella as well. Bacteria can ferment glucose with acid and without H2S. Production of gas is variable. Utilization of lactose can be possible in the case of Shigella zona, but mainly bacteria is very inert biochemically, and more than 1 day is required to identify good biochemical properties of microorganism. So in routine practice, biochemical identification of Shigella is rarely used, and the main test which is used to identify Shigella, it is a serotyping according to O antigen. And multiple sera can be used, monovariant, polyvariant, and the same species of Shigella can be identified as a serogroup. So we have group-specific serum which can recognize Shigella dysentery, and if you will use serotyping, it's more correct to say that it is Shigella serogroup A. Shigella flexneri is Shigella serogroup B, Shigella boydii is a serogroup C, and zona is group D. All Shigella except Shigella zona have subclassification into type-specific O antigen group, O antigens. And we know that in nowadays Shigella dysentery is divided into 15 serotypes, Shigella flexneri into 19, and Shigella boydii into 20 serotypes. But again, according to different data, different countries, different laboratories, those numbers can differ, and some say that it is 17 serotypes of Shigella dysentery. So actually in medical practice it's not really important. The key is to identify Shigella dysentery type 1 and species or serogroups of Shigella. Shigellosis is the common name for disease caused by Shigella except bacillary dysentery. It is extremely important disease worldwide, and more than 165 million cases of disease are registered annually worldwide. Disease is a real problem for multiple governments and for some world organizations. Disease is transmitted human to human from ill or carrier person. Few cases from large primates were described, but it is more like an exception. Of course, disease is transmitted by fecal-oral route from one person to another, or by contaminated food or water when hygienic level is very poor, or some sanitary conditions are extremely bad. Incubation period is short, not more than 4 days, and next week subject will have common symptoms as a fever, malaise, and diarrhea will appear. Usually shigellosis manifests with watery diarrhea, maybe with mucus but without blood, cramping, pain in abdominal cavity, and the key changes in intestine and pathognomonic changes is a destruction with enterocytes, with formation of ulcers and colitis in severe cases, especially when it is dysentery. So if we'll talk about pathogenesis and infectious cycle of Shigella, it is very complex and difficult with multiple steps. I will show you only key stages of the disease just to understand how disease can develop. So in routine shigellosis, if usual shigellosis, bacteria will be ingested with the food or water, and will get into the intestine, into the colonic epithelium, and via M cells it will penetrate in the laminal space. So M cells will engulf Shigella by formation of endocytosis, and bacteria will be delivered to tissue macrophages and engulfed by macrophages. But actually, due to production of several factors, bacteria can resist phagocytosis. It will escape from phagocytic vacuole and will induce apoptosis of macrophages, which will be destroyed, and bacteria will be released in the internal part of intestine already. So damaged macrophages, damaged M cells will make it more easier for the rest Shigella to invade in the luminal space of intestine, and here Shigella will be uptaken by epithelial cells. Epithelial cells will form like a micropenocytic invaginations. They will engulf bacteria. It's like a podial extensions, and bacteria will invade into the enterocyte. Here again, bacteria will remain alive, it will multiply, it can move within one cell or even can shift between different cells. And in destroyed cells, destroyed macrophages and affected enterocytes will release number of cytokines. Interleukin 1 and 8 are the most important. They will induce chemotaxis of leukocytes and other macrophages, and some of them will lead to destruction of epithelial barrier, destruction of our tissues, and will make disease only more severe. So neutrophils will need to invade into the intestine as well, and they actually can destabilize the junctions between epithelial cells and make intracellular space even more wide, and other Shigella which were still in intestine will be much easier to invade into the epithelium, and it will worsen in the disease. So here bacteria will release enterotoxin, which will affect excretion of water and minerals, and watery diarrhea will appear. So if it is regular shigellosis, then disease is self-limited. In general, more number of macrophages and leukocytes will eliminate the infection, and disease will progress to recovery. But as we already said, some Shigella, Shigella dysentery type 1, serotype 1, can produce shigatoxin, which is a cytotoxin, and disease will not finish, it will progress. So release of shigatoxin will lead to absorption of the toxin in the blood circulation, and Shigella will still remain in the epithelium of intestine, but toxin will be absorbed and affect overall blood capillaries. Toxin of Shigella, as any other biological toxin, consists of two main subunits. It is A and B. A subunit is active, consists of two subunits which are fixed to a pentamer of 5 B subunits. B subunit is binding. It is responsible for recognition of unique receptor on the surface of endothelial cells. It will fix and allow A subunit, active subunit, to invade into the cell. So in general, the toxin will attract platelets. They will adhere to the endothelium of blood vessels. Blood vessels will be obstructed from the internal part, and it will lead to hemolysis of red blood cells. They will need to squeeze through that restricted space, and they will be damaged, they will be destroyed, and in general, the disease will have systemic manifestations. All symptoms in the dysentery can be divided into two main groups. It is intestinal symptom and extraintestinal symptoms. So in intestine, diarrhea, which initially was watery and profuse due to release of enterotoxin, will be changed with the less frequent diarrhea, and water will be changed with the blood. Volume of stool will decrease, and now it will be stool with the blood, with mucus, a lot of leukocytes here, and of course some pain will appear in intestine. But again, toxin have activity not only against intestine, it can damage blood vessels in the whole body, and two most frequent complications of bacillary dysentery is hemolytic uremic syndrome and reactive arthritis. Again, we talked already about hemolytic uremic syndrome. It is the damage of kidney capillaries with acute renal failure, hypertension, and severe toxic symptoms. Can progress to severe renal insufficiency and even acute renal failure with the lethal outcome. Reactive arthritis is less dangerous complication. It's rarely observed in people after bacillary dysentery, but again, it's characterized by painful joints, especially joints of fingers. It can be associated with painful urination and irritation of eyes, which is known as Reiter syndrome. And in general, bacillary dysentery without proper treatment can have mortality rate till 15%. So simple shigellosis, when it is just infection without production of shigatoxins, do not require any antibacterial treatment. Routinely replacement of lost electrolytes and liquids is enough. But if it is dysentery, then of course you should remember that dangerous complications are possible, and antimicrobial treatments such as ampicillin or cephalosporins can be and should be prescribed for affected person. Common measures of sanitation, of course, should be used. Isolation of person who is confirmed with shigellosis until complete recovery. You always need to identify that person who infected your patient, because disease is person-to-person transmitted, so it is obligate to find out the source of infection. If it is a carrier, then eradicate carriage. And of course, the main task for government of the country is to provide proper sanitation measures. We have that famous company fight with street toilets in India. It was kind of funny for routine people, but for medical person it is obvious that measure is extremely important, and it is the key to fight that serious infectious disease. So, few manifestations of that reactive arthritis, different joints can be affected, different severity of the symptoms. It can be a weak manifestation, but in some cases can be quite severe, and it can affect regular motility, regular life can be affected due to the disease. So, officially we have no licensed Shigella vaccine on international level. In Russia we have our own vaccine, it's known as a Shigella. It is a polysaccharide vaccine of Shigella zone. It can be used as an injection and provides immunity for about 12 months. Usually we recommend that vaccine for people who are going to travel to endemic countries, or for laboratory workers, or those people who have high risk of contact with Shigella. Immunity will appear in 2 or 3 weeks after injection, so that vaccine should be planned some month before trip. Vaccine is contraindicated for kids below 3 years of age, so that group of people remains not protected. So we have a lot of vaccines which are still in research and development stage, multiple different options. It is a conjugate, subunit vaccines, whole cell, and some antigen vaccines, but all companies who are designing those vaccines, they have no stable promising results. Recently, India government announced that Shigella vaccine is developing by India as well, and preterm results of experiments show very promising data. And officially it was stated that India is going to design and license good Shigella vaccine till 2024. Maybe it will be so, and if it is really so, then India will be the first country who will design that vaccine, and maybe very soon we will have some new results according to that vaccine. So that's all for current lecture. See you. Bye bye.